Welcome to MusB Research Cell Culture series. Today we are going to talk about general introductions of cell culture, types of circle cultures and how they are done in lab and what precautions be to take during cell culture and what kind of application cell culture can have in lab and industry and in your studies. In this video, I'll walk you through what cell culture is, why it matters, and the key principles you need to follow in the lab. Let's get started. What is cell culture? Cell culture is the process of growing cells outside their natural environment, usually in a laboratory setting, under carefully controlled conditions. These cells can come from plants, animals, or humans. Once they're isolated from tissue, they're placed in a culture flask or dish, provided with nutrients, and maintained at the right temperature and atmosphere so they can survive and grow. This allows scientists to study cellular behavior in a controlled and repeatable way. Cell culture is a core tool used across many areas of research. It plays a major role in drug discovery, where new compounds are tested for safety and effectiveness. It's used in toxicology studies to evaluate the effects of chemicals. Researchers also use cell culture to model diseases such as cancer and viral infections. Cell culture is essential for vaccine development, diagnostics, and understanding basic biological processes. There are different types of cell cultures, and it's important to understand their differences. First, we have primary cells. These are cells taken directly from tissues. They closely resemble cells in the body but have a limited lifespan and stop dividing after a certain number of generations. Then we have cell lines, also called continuous cell lines. These cells can divide indefinitely and are commonly used in research because they're easier to maintain and provide consistent results. Cells can also be classified based on how they grow. Adherent cells attach to a surface, while suspension cells float freely in the culture medium. For cells to grow properly, they need very specific environmental conditions. Most mammalian cells require a temperature of 37 degrees Celsius, a carbon dioxide level of about 5%, and controlled humidity. These conditions are maintained using a specialized incubator. Equally important is sterility. Even minor contamination can affect cell health and compromise experimental results, so maintaining a clean environment is critical. Cells grown in culture depend entirely on the culture medium for survival. Choosing the right medium for your specific cell type is one of the key steps in successful cell culture. One of the most important aspects of cell culture is aseptic technique. This means working in a biosafety cabinet, wearing gloves and lab coats, and using sterile tools and reagents. Surfaces and equipment should be properly disinfected before and after use. Following these practices helps prevent contamination from bacteria, fungi, and mycoplasma, and ensures both sample integrity and personal safety. Even with good technique, problems can still occur. The most common issues include contamination, poor cell growth, and unexpected cell death. Regularly observing cells under the microscope helps detect these problems early. Maintaining good laboratory habits and responding quickly to changes in cell behavior can prevent small issues from becoming major setbacks. To sum up, cell culture is a fundamental technique that supports much of modern biomedical science. By understanding the basics and following good laboratory practices, researchers can generate reliable results and contribute to meaningful scientific progress. If you're working in the lab, mastering these principles is an essential first step in your research journey. If you found this video helpful, feel free to like, share, or subscribe for more science-focused content. Thanks for watching, and stay tuned for next video on this cell culture series. Please leave your comments if you would like to see a video on specific topic related to cell culture, microbiological culture or in other culture technologies.